Okay, biochemistry, now we get to talk about proteins. You should be very excited. This is very awesome. This is what biochemistry really is about. Okay, so part 1, we're going to talk about the amino acids. And you're going to have to draw and name all 20 common amino acids. So I say common here, they're also referred to as the natural amino acids. There are unnatural amino acids, such as selenocysteine, selenomethionine, etc. We're not going to be concerned with that. This is mostly for the exams. You need to have some general idea of what amino acids look like when they're mentioned in problems. And this really just comes from experience. So I suggest that you draw this out, as many times as you possibly can. Additionally, it's going to be important to categorize the amino acids into some categories that biochemists have agreed on. Nonpolar aliphatic, nonpolar aromatic, polar, positively charged, aka basic, and negatively charged, aka acidic. Additionally, you're going to have to identify unique properties of amino acids, such as ability to form salt bridges, disulfide bonds, hydrogen bonds, hydrophobic interactions. So each amino acid is going to bring a unique property to the protein that it's part of. So it's important to have a really good fundamental understanding of the amino acids. So that's what this is about. Okay. So what I've drawn for you here is the basic amino acid. So here we see the part that's the amino functional group and the acid functional group. Here is a hydrogen attached to the alpha carbon. Also attached to the alpha carbon, I've written the letter R in red. This is the side chain. So there are 20 different side chains. Well, at least 20 different side chains that you need to be concerned with. Okay, so the first group of proteins that we're going to talk about are the nonpolar aliphatic. So what this really means is that these side chains have carbons and hydrogens, and that's it. So that's what nonpolar aliphatic means. Aliphatic just means that it's not aromatic. Okay. So glycine. So and I've written here, this is the full name of these amino acids. Here's the 3-letter abbreviation. Here's the 1-letter abbreviation. Memorize this. Really, this is just from working it out, writing it out, practicing. Okay, so for glycine, this is really interesting because the R group is just a hydrogen. So this is really cool, right? So if we draw this out, CCOOH, NH3+, then there's always an H. Glycine has another H. Therefore, is this carbon chiral? Answer that for yourself. Does it have 4 different attachments? Okay. Next up, alanine. R group is CH3. So if you're drawing this, CH3 would go here. I hope you get the picture. Valine, the R group looks like a V. It's 2 methyl groups, CH3, CH3. Okay. Leucine and isoleucine are a little more difficult because their structures are very, very similar. Here we go. Leucine looks like this. It's like you had just added another methyl to the valine group, whereas the isoleucine looks like this, where you move this methyl group to this position. Okay. I'm not going to draw all these out down here. Something I would like to note before we move on is that most amino acids are in the L form. So we would see things like L-glycine. This is interesting and opposite of carbohydrates. Most carbohydrates are in the D form, so it would be like D-glucose. And that's because, just briefly, the origin of amino acids is believed to be from meteorites, so they came from outer space, whereas the origin of carbohydrates is from this planet, so those came out as D, and just in space it became L. Interesting, right? So interesting. Okay, so up next, let's talk about some nonpolar. These aren't technically aliphatic, although many people consider these two also in the nonpolar category. Okay, so first we've got Prolene, abbreviated PRO and P. This is the only amino acid. Amino. Because as you can see, this structure is different. The R group branches off and goes back and reconnects with the amino group. So Prolene and Glycine cause the two smallest amounts of space. So we see these a lot in proteins that have beta turns and need they don't take up a lot of space. They cause bends and kinks. Prolene and Glycine do that. Methionine is another interesting nonpolar one. As you remember from Biology 115, this amino acid is encoded by the codon AUG, which is the start codon. So every single protein, as far as you are concerned, starts with methionine. And this is what it looks like. Interesting is this sort of ether type bond with the sulfur. Actually, if this was an oxygen, you would consider it polar, but because this sulfur, this is a sulfur and not an oxygen, this whole side chain is actually nonpolar. So that's something to remember. That's one of the differences between sulfur and oxygen. Okay, up next we're talking about nonpolar aromatic side chains. So we see there's only 3. And the importance of the aromatics is that these contribute to the UV visible wavelength of each protein. And because the numbers of these are different in each protein has a unique extinction coefficient at 280 nanometers. So the optimum wavelength for proteins is 280 nanometers. But the extinction coefficient is unique to each, and it's based on the number of these residues, because these are the aromatics. They absorb light in the UV visible range. Okay, so phenylalanine, we notice that this has the word alanine in it. This R group, if you remember, alanine is just CH3. There's the CH3, and then a phenyl group attached to it. Phenylalanine. Tyrosine looks just like phenylalanine, except it also has a hydroxyl group para to the alanine portion. And I would also like to point out the abbreviation here. This is Y. This is not a T. Several amino acids start with T, so you have to really pay attention. Also, phenylalanine doesn't start with P. We've already used that for Prolene. It starts with F, because the sound phenylalanine starts with F. Interesting, tryptophan is W, because this has 2 rings in its structure. This is kind of fun to draw in this tiny space. Forgive me. So 1 5-membered ring, 1 6-membered ring, double bond in between the 2 rings, and then fill out the rest of the double bonds so it's aromatic. Boomski. Tryptophan, because it's got 2 rings, so that looks like a W if you just kind of trace like that. Ha ha ha. So fun. Okay, coming up next are the polar amino acids. Okay, so serine, SERS, cysteine, CYSC, threonine, THRT, asparagine. I would like to point out that this is ASN, not ASP, and its abbreviated 1-letter abbreviation is N. Similarly, glutamine is GLN and Q. So this is kind of, they were running out of letters, right? That's pretty much what happened. Okay, let's go into the R groups. So a lot of these have either amines or hydroxyl groups. So serine, the R, looks like this. Cysteine is an interesting one because this has a free SH group on the end that can form disulfide bonds. Let me put a star and say disulfide bonds. That's very unique. So disulfide bonds, we're going to see, are going to be used to stabilize the tertiary structure of a protein. So this looks the same as the serine, but instead of OH, SH. Threonine looks like serine, but it's a T. And remember, there'd be another H here, but this is CH3. This is OH. And now we get to do the funky ones, asparagine. 1, 2, double bond, NH2, because N likes 3 bonds. I have to remind myself, right? And just a reminder, this is where the alpha carbon would attach. And then finally, glutamine, same as asparagine, 1 more carbon. 1, 2, 3, I'm out of room. Double bond O, NH2. These are our polar ones. Okay? Now we can move on to the last 2 categories, which I'm going to write together, positively charged and negatively charged. So histidine, HISH, lysine, LYSK, arginine, ARGR, this is the pyrite amino acid, aspartate, ASP, different than ASN. So this is aspartic acid, or aspartic acid, so aspartic acid, there you go, with a D. Glutamate is the glue, GLU, and its abbreviation is E. Okay, so let's write these R groups. Histidine is a pain, but you have to remember that it's in a resonant form. 5-ring structure, and then you can distribute the ends. One can go here and one can go here, or one can go here, one can go here, it doesn't matter. And then with the double bonds, we would see that this is NH. But because it's resonant, these and the lone pairs, these bonds are constantly moving. So it's kind of harder to draw, but it's easier because there's so many correct ways to draw it, so keep that in mind. It's not aromatic. If you count up the lone pairs and the double bonds, this is not aromatic, so keep that in mind too, even though it looks like it could be. Lysine. This is a long one. This it's connected to the alpha carbon here, and then it has 1, 2, 3, 4, and then an NH3+. Arginine's another long one. And I hope I have enough space. 1, 2, 3. 3 carbons, then NH, and then a carbon that's attached to double bond NH, NH3+. Right? This one is really sort of unique and bizarre because it's got 3 nitrogens in it and a double bond. Very, very interesting. Okay, and then on to the negatively charged. You're going to notice that the aspartate and the glutamate have the same number of carbons in general structure as asparagine and glutamine, except instead of these NH2s, they're going to have OHs. So they're really, really similar. So R, double bond O, O minus. Glutamate has 1 more. 1, 2, O, O minus. Okay, so as you can imagine, based on these structures and drawing positive and negative together, if we stack the positively charged and the negatively charged, we have this great ionic interaction, and this is one way that proteins fold. Additionally, we can bury the hydrophobic, these nonpolar amino acids, Anything big and branchy needs to be buried away from the water, because when water molecules line up with something hydrophobic, it creates a decrease in entropy. So that is not good for the overall thermodynamics. The polar residues, so these are nonpolar, those would be buried. Oh, additionally, before I talk about polar, the aromatic residues can stack on top of each other like plates and cause stability that way because of their overlapping pi orbitals. The polar residues like to be near water. They're really great at forming hydrogen bonds. So these are typically found on the outside of a protein that's exposed to water. These can also line the active site, as well as the positively charged and negatively charged. And a lot of times that these can be used for chemistry. The polar and the charged residues can be used for chemistry within an enzyme. So keep that all in mind. Okay, I think that's pretty much covered everything. The last thing on your list is explain why glycines chiral and nothing else is, or is achiral and nothing else is. And it's because of its R group, right? I asked you that in the beginning. So glycine's the only one where if you draw it out, this R is an H, which means that the carbon has two of the same attachments, therefore it's not chiral. So it's really, really unique. It makes it very small, good for turns, along with Prolene.